So let's talk about why use electrons. Well, if we wanted to build a microscope, there's a number of different kinds of radiation that we could use, and each one has its advantages and disadvantages. So the first one you might think of is visible light. And the advantages of visible light are, first of all, it's not very damaging. Obviously, we've evolved to live bathed in sunlight. And you can image cells in a light microscope for many minutes without damaging them much. If you illuminate them strongly, eventually you'll see the organelles slow down, and it is damaging, but it's much less damaging than the others that we'll talk about. In addition, visible light is easily focused. All we have to do is take a piece of glass and shape it right, and it will focus visible light. And finally, our eyes have evolved to be wonderful detectors for visible light. But there are some disadvantages to visible light. The main disadvantage is the wavelength. The shortest wavelengths of visible light are approximately 400 nanometers. And as we'll discuss, a fundamental limit to the resolution of a microscope is the wavelength of the radiation used to form the images. And so 400 nanometers is too large to see many of the ultrastructures of cells that we'd like to. So if the primary disadvantage of visible light is the long wavelengths, you might ask, well, why don't we just use shorter wavelengths of light? And of course, some of these are called X-rays. And X-rays have the advantage that they can have a very small wavelength. In fact, for instance, the X-rays used for X-ray crystallography have wavelengths of 1.5 angstroms or even smaller. And they also have great penetration, and that's why, for instance, you can go to the hospital and have an X-ray image taken through your entire body, because they'll pass through your entire body. But there are, again, strong disadvantages. The main one is that X-rays are hard to focus. It turns out that glass doesn't have a high enough index of refraction to focus X-rays effectively. And in fact, we don't have a good material to focus X-rays effectively. Instead, people use what are called zone plates, but they have their own technical difficulties. And so X-rays are hard to focus. Next, X-rays damage the sample. And that's why, of course, if you go to the hospital for an X-ray, they'll give you a lead blanket to cover the parts of your body that don't need to be imaged. So let's think about other forms of radiation that we could use to build a microscope. And as soon as it was discovered that electrons also have a wave-like property, it was realized that the wavelengths could be made very small. In fact, the electrons that we typically use in our electron microscope at Caltech, they're accelerated to 300,000 volts. Their wavelength is just in the picometers, orders of magnitude smaller than a single atom. And so one of the great strengths of electron microscopy is that the wavelength of the radiation being used is exceedingly small. It's not the limiting factor, even close to the limiting factor. Now, the other advantage of electrons is that they can be focused, and we'll talk in some detail about how to focus electrons. But there are also strong disadvantages. First of all, like X-rays, electrons damage the sample terribly. And so a fundamental limit of electron microscopy is the radiation that a biological specimen can sustain before its structure is degraded. In addition, electrons also have very poor penetration. So another fundamental limit of electron microscopy is that we can't effectively image samples that are thicker than about 1/2 of a micron. So while we're thinking about different radiations, we might consider, is there anything else that we could use to make a great microscope? And there is one that should be talked about, and that's neutrons. Could we make a neutron microscope? The advantages are that neutrons would produce very low sample damage, and neutrons can be made with very small wavelengths, just like electrons, even in the picometers. So that would be fantastic. The disadvantages, however, are, first of all, how do we produce a bright beam of coherent neutrons? We simply don't know how to do that yet. And also, how do you focus them? The best ideas would be to use the zone plates again, but there are technical limitations to the zone plates. So this is why an electron microscope fills a special niche in the microscopes that could be used to investigate cellular structure. And this brings us to what I've called the structural biology continuum. Now, usually when we say structural biology, most people think of the technologies X-ray crystallography and NMR spectroscopy, which are used to solve the structures of proteins, sometimes individual protein domains, and they give us atomic models. But as proteins, for large proteins and for protein complexes, it can become more and more difficult to crystallize them. And NMR has other technical limitations that prevent it from being used effectively for large proteins in their complexes. And this brings us to the realm of where electron microscopy is most effective. Electron microscopy allows us to image large proteins in their complexes. We call this technique cryo-EM single particle analysis. And in addition, X-ray crystallography is becoming more and more powerful and actually delivering structures of larger and larger complexes. So it plays an important role in this arena. Now, we've discovered that most proteins actually act as part of assembly lines within the cell. And so to understand the relative positions of these proteins, which are dynamic and can be at larger distances, we need to be able to image multi-protein reactions. And we can do that with electron microscopy, both in vitro and also within cells, as we'll talk about later, through cryo-electron tomography. Now, at even lower resolution, we get to the realm of light microscopy, which allows us to also see the dynamics of how individual proteins move around within the cell. Now, on either end of the structural biology continuum, I add two different computational techniques. First of all, systems biology, or structurally and spatially explicit cell modeling, can allow us to explore different hypotheses for the rules that govern how proteins are interacting and where they can go and what they can connect with. And systems biology allows us to explore that computationally. Now, on this other end, I put the computational technique molecular dynamics simulations, which, if given a model of the positions of all the atoms in a protein, molecular dynamics simulations allow us to explore models and test how the positions of those atoms allow the proteins to perform the functions that they do. And so together, all of these techniques are needed to understand cells at a structural level.